I'm talking to people who have challenged the status quo or challenged a paradigm in some way and really called our attention to the fact that the research wasn't there, or artists who have suggested a different way of doing things. And the goal for that is to really sort of inspire primarily younger people to sort of think a little bit more critically. Imagine that it's not just the way you're being told. So when Seafried told me about your work, I was like, oh my God, I have to have him on the show. And then we probably need to bring you into all kinds of other broken science things happening. But just to get started, I feel like I have, let's see, 123 pages of notes on you. So the show is not the place to do like a super deep dive into all this stuff, but I actually would love to talk to you about writing for us or contributing in some other way, because this story is, I mean, it's a movie. It's incredible.
Just to let you know, I'd have to tell you that probably 4 years ago, I mean, and so much has happened in the past 4 years, but 4 years ago, a guy who is I'm going to call it a videographer, but who worked with some really big name people on documentaries, very big name people, the biggest actually, came up with the idea that he wanted to do a documentary on my story.
Great.
And he came out and they spent 2 or 3 days interviewing me in many different facets. And as it turns out, nothing ever really came of it, because the videographer who had the connection ultimately became, vice president of some other, company. Oh. It's kind of like the trailer type material, if you're going to do a documentary, and then it just kind of was just left. But there's been so much discovery since then. And also something weird happened yesterday that you should know about, because it relates to this in a general sort of a fashion. I'm pretty big in the area of this world called Hormesis. I'm like Mr. Hormesis, actually. Recently, I wrote a couple of papers, substantial papers based on Hormesis, how long can we expect to extend at maximum human lifespan. And it's a really substantial paper. And as it turns out, then I got interviewed on somebody's podcast, was seen by lots and lots of people. And as a result of that, there's a group that does documentaries and movies, and they contacted me yesterday, and they're putting together a documentary on longevity or lifespan and stuff. They want me to be part of their documentary. Like one of the dozen or so people that how you interview people that are supposed to know something, and so I'm an expert that they would talk to.
That's great.
One is the LNT story we'll talk about, but the other is this Hormesis story, which relates to LNT, and it's how did the scientific community get the entire concept of dose response wrong for the whole 20th century. And now my thing, which is Hormesis, you won't believe this, but Hormesis, which was within the last 4 years, they've identified 8 hallmarks of health. And Hormesis made one of the 8 hallmarks of health. I mean, that's unbelievable. And my concept has gone from the outhouse to the penthouse in 25 years. There are a couple of revolutions going on here, and there really needs to be a light that's shown on them.
Yeah.
But they are different. And the Hormesis one will revolutionize Alzheimer's disease treatment and Parkinson's treatment and diabetes treatment and longevity issues and plant growth. It's not just the humans, it's plants. It's anything that's alive that has to have a dose response. Hormesis will be there. And the scientific community actually missed it. And I've figured out and written about why they missed it and why they continue to miss it.
So let's start there. I mean, I feel like that's a great place for us to just kick off. You could introduce yourself, because I think that is really important to the sort of looking at radiation and cancer and mutation and all of these other things that you have, I would say, sort of single-handedly unraveled from my perspective.
I hate to say this, but it's probably true. It's probably true.
And I think, you've gotten some attention for it recently, but the truth is you've been working on this for a long time. And I mean, you've done really what as a journalist, some incredible primary research, gathering letters and finding correspondence to really fill in the gaps to help explain some of this stuff. So I want to do something a little bit differently than I usually do, which is We'll start by I'm going to ask you some questions, and I would like to talk about hormesis because I think it's actually really critical to understand that that's your background, so
Sure.
that when you were looking at the model, you could see this is sort of like counterintuitive to the way that we know, mammalians respond, humans respond to toxins. But I also then I'm going to kind of turn it over to you because I think there's so many twists and turns in the story that I don't want to mess it up. And so usually I would lead the person through the story, but I think in this case, it's so involved that I'm going to kind of let you just tell it, and then I'll interrupt you and ask for clarification or significance along the way, if that sounds good.
Sounds good to me.
First of all, why don't you explain what hormesis is?
Well, hormesis, which is my offspring, so to speak, is really a dose response relationship. That's how I define it. And it's a dose response relationship which is characterized by a low dose stimulation and a high dose inhibition. It's a biphasic dose response relationship. And that's kind of different than the way I was brought up to learn about dose response relationships. I was brought up to believe, more or less, that the predominant ones were either a threshold dose response in which nothing really goes on at low dose, and then you kind of pass a little zone, and then an effect happens. It's kind of like drinking alcohol, and that is if you've either consumed too much alcohol or alcohol too quickly, then you start to feel a little bit lightheaded, high. That'll show either a dose response relationship or a dose time relationship, dose rate relationship. But there is a threshold. You have to pass a certain threshold before you actually experience an effect. That's the threshold type model. The other is a linear model, and the linear model has been until recently only applied to mutagenic carcinogens, genotoxic carcinogens, and this is a linear dose response which argues that the response is directly proportional to the dose. And so what that really means from a carcinogen point of view is that that there is no dose that's safe, that even a single molecule or a single ionization from radiation could start the process going. So as a child, for example, when I was about 13 years old, I smoked 2 cigarettes to try it out. Okay. And I'm saying, oh my goodness, I smoked 2 cigarettes when I was 13 years old. That couldn't be a risk for anything, right? But the linear dose response model say, oh no, you increased your risk of getting lung cancer or all the other cancers that are associated with smoking as a result of what you did when you were 13, when I snuck 2 of my father's unfiltered Chesterfield cigarettes.
Delicious, I'm sure. You love that experience.
I only lasted through 2, right? And so, and thank goodness I never went back to it again, right?
Right. I tried smoking a cigar at around the same age and didn't know that you weren't supposed to inhale and threw up everywhere. So I had a similar experience.
Well, as it turns out, for me, the idea, I never even knew that the term hormesis until 1985. But for me, the concept of hormesis started a lot earlier. When I was a mere child of 20 and taking a junior in college. This was 1966. Okay, so way back when, I was taking a plant physiology class, and the professor overburdened us with probably 40 different experiments to do in a greenhouse and in a laboratory. We had to write every experiment up like it was a scientific paper, and it was a massive amount of work. And one of the little experiments that we had to do was a dose-response relationship, and it was to treat peppermint plants with a synthetic growth inhibitor. And it was really just to teach us what a dose response was and to just get a feel for how to do a particular set of experiment. And so I remember this, and it should have been a dose-dependent decrease in growth. The more that you give the inhibitor to the plants, the less they grow. It's just a dose-response relationship showing, and it should show most probably a linear dose response inhibition or slowing down of the growth. Well, as it turns out, this big burly guy comes into our classroom in November, cold day in November '66, and he says something's going wrong with this peppermint study. The plants are being stimulated, and it never has happened in the last 5 years that we've done this exercise. And so something's going wrong. So I don't know what's going wrong. Maybe you used the wrong chemical, you mislabeled something, you did something that was different. I don't know what it was, but these plants are being stimulated. And so he said, I want to find out why. And he said, it's too late in the semester to figure it out. He said, but if any of you in the class, and there are about 24 of us in the class, has any interest in working with me to figure this out, then come back and see me after the semester's over. So as you can figure, there was one person in the class of 24 that had an interest, and it happened to be me. So I go back and I see the guy and I say, I'd like to figure this thing out. And so he said, well, we'll start by trying to figure out what you did, and then we'll replicate what you did, what I'm telling you to do, and we'll see how it all comes out. Well, in going over how I made the stock solution for the original classroom experiment, we determined that I had made an error in making up the stock solution, and I made it up such that it was about tenfold more dilute than it should have been. So I calculated what I was supposed to have done incorrectly, and so he said, well. He said, then let's go back and do what you did, and we'll combine what you were supposed to have done and do it correctly, and you'll have a bigger experiment with more doses and so forth. So I went and I followed that lead, and after the experiment was over, I found that, yes, indeed, at the higher doses, it inhibited exactly like he said they would. The plants are more compact, darker green, they were impressive. But at the lower doses, the plants were stimulated. And it showed what I'd call a biphasic dose response with a low dose stimulation and a high dose inhibition. And he said, and I said, well, this took weeks to do. And so I said, well, I think we answered the question. He said, you think you've answered the question. He said, but in science, he says, you have to replicate your work, and you have to replicate it, and you have to prove not just to yourself, you have to prove to others that are very critical that you have enough evidence, for the statements that you're going to make. That's what a scientist actually does. And so you have to go back in and you have to replicate that again, and you actually have to add more plants and more doses to what you have. You have to make it stronger and go back and do it again. Now you know the basic plan. So I went back and I did it again, and I got the same results. And I said, well, it's over now. And he says, no, it's not over now. You think it's over. It isn't. He said, I have to tell you that your stimulation that you think is real, you're getting only about a 30 to 50% increase in the growth over your control group. You don't know enough about this peppermint plant to know what the background variability is. It could be, yes, you're getting this 30 or 50% over the control group, but he said, you have to know more about the background variabilities, statistical variance, everything to ensure that that's a real treatment effect. You have to go back and replicate it again, maybe more times than you might imagine, to see just how consistent your findings are. And I'm saying, wow, this is what science is really about. I'm this young guy, but getting my first taste of, being actually, there's excitement, and then there's the actual hard work and the drudgery. There's a lot to it, and the people don't really know because they just look at the data at the end of the road. But there's a lot in between. And so this guy, in his forcing his will upon me, made me replicate it about 12 times. 12 times. And I went into him one day frustrated, and I said, I don't care whether you believe this or not. This is reproducible. You can replicate it. Anybody could replicate it. If I can do it, I'm done with this. And he says, I agree. He says, you've proven your point on this. He said, but it's raised some new questions. And I said, well, what new questions could there possibly be? He said, well, what you've done is you have shown that applying this solution to the soil affected the growth of these plants. Low dose stimulation, high dose inhibition. We don't know why, but it did. He said, maybe this is happening because there are microbes in the soil that are metabolizing the chemical to some new form of the chemical, and it's this new form that's causing the stimulation. So I think what you should do is do the exact same experiment over, but this time grow your plants in hydroponics. Get out of the soil matrix, and that'll you will learn something from that experiment. And
Well, he's teaching you to isolate the variables, right? So that you can actually make conclusive.
yeah, and so I said, well, that's really a good idea. That's a. I wished I had that idea, but I didn't. And so, but I had to learn how to do hydroponics, and that was good because that was a whole different dimension. And so I ended up doing that. It was a lot more work doing hydroponics, but I ended up replicating. I got the same results. I got a low dose stimulation, a high dose inhibition. He made me replicate it about 6 times, and and at the end, I said, wow, I said, I think that what we've discovered is that this chemical acts in a biphasic way. I never knew the term hormesis. I looked in a lot of literature, never related it to that. We published the study, and we published it in a British plant physiology journal, and I referred to it as a low dose stimulation and a high dose inhibition, never ever relating it to the concept of hormesis. This guy was so demanding on this young student at the time that I just said, I don't want to go. I love biology, but he burnt me out on plant research. And at the same time, it was the beginning of the environmental revolution, Silent Spring, Rachel Carson. I had been involved in another project dealing with pesticide exposure on Cape Cod.
Pause for 1 second, because I think part of what you're explaining in great detail, I think we need to like sort of back out a little bit and explain to the average person that part of what this is this notion that things that are perceived to be toxic or harmful at low doses can actually be helpful or healing. And that this is revolutionary in the sense of, and sort of in trying to think of an analogy or a way that, the kind of average non-science person might understand what you've revolutionized. There's this sort of phenomenon in psychology and child rearing that's like the growth mindset, right? And it's this idea of giving kids little failures, little points of, negative experiences so that they learn to be more resilient and they can take on challenges and they become more confident over time. That's that theory. And I think part of what you've discovered or what you've been able to prove so well in a wide variety of things, which we'll get into, is that the human body responds in a similar way. That there are toxins that we know are toxic. And, like eating an apple seed a day has a little bit of arsenic in it that might be actually beneficial to you. The threshold model is basically saying there's no interaction below this point. Above this point, it's all negative. Below this point, there's nothing. What you're saying is actually below this point might be a good thing, not a nothing thing, not zero, right? It doesn't go from a reaction to nothing. And the linear model suggests that all the way down, you're going to have negative. Is that a fair characterization?
Yes, it is. And I can share with you how I made that transition to the audience, if you'd like.
Yes.
Okay, so that's kind of how I left it more or less as this young budding scientist, and went on and changed my direction and became an environmental toxicologist, got my PhD, began my professional career as an assistant professor, and ultimately at the University of Illinois at their School of Public Health. And then I came to the University of Massachusetts in 1976 and tried to make my name as any young assistant professor would, and took advantage of, as I said, the environmental revolution with funding grants and developing my expertise in the area of toxicology. And then after I'd been here about 10 years, and which is really now 20 years after I was this undergraduate student, I got this brochure in the mail. And the brochure was about this concept and this term I had never heard before. It was about radiation hormesis. And so I for whatever reason, I read the brochure, and it talked about this strange thing that at high doses, radiation was very harmful, could cause cancer, could cause a lot of other effects. But at low doses, it might actually be able to reduce the cancer risk and cause other benefits. Wow, I said, that's interesting. And then I said, that's a biphasic dose response. I said, that brought me back 20 years.
The peppermint plants came back to you.
Yeah, yeah. In fact, they did. I said, I've seen that before in my work when I was an undergraduate. I said, I said, the weird thing here is that biology is biology. And I said, I know that this biphasic dose response exists and it occurs and it was very repeatable. And I have this massive amount of work because this guy worked me to the bone, so to speak. And I said, this may actually prove to be enlightening to these people who are studying radiation. I didn't have any great knowledge of radiation. So what I did was, this was a transformative step in my life. I got on the telephone and I called this person up who was directing this conference. And Leonard Sagan was the medical director of the Electric Power Research Institute in California. He asked me if I had a difficult time getting my paper published. It's an interesting question to ask. And I said, well, strange that you asked that because it was the very first paper that I ever published in my life. I said, I submitted that paper and I got it rejected 6 times in a row by 6 different journals. And he said, well, they biased against your what I'm going to call, he said, your hormetic dose response, because he felt there was a lot of bias against hormesis in the scientific literature. And I said, well, in all honesty, I said, I was pretty incompetent at that stage of my professional life. So it's pretty hard to separate one thing from another. But I can tell you for sure that every time that I got rejected, I looked at the reviews that were written by the reviewing group, and I tried to learn from them. So I got the paper published in a really good journal, but it was a struggle for me to do it the first time. And that was way back when, I said, and I know the game today because it's now 1985. And he said, well, I'd like to invite you to come to our conference and give a talk. He said, but you have to develop a paper on chemical examples of hormesis. The conference will all be about radiation, but I want the people to see that this can be extended in a much broader sense to the world of chemicals. And so that's actually how I then transitioned into begin to, wow, they're looking at all sorts of health effects that range from extending life, reducing cancer, reducing all other kinds of incidents of disease, and so forth. And it opened my eyes to not just plant hormesis, but animal hormesis and things that could begin to affect health and a whole range of biological performance. And that was in 1985. And I'd have to tell you that despite the meeting and everything, I was not very excited because I was well funded. When you go in a certain area, it's only important to you if you can get funding in that area. You can't just follow.
When your work at the time, the funding sources were looking at hormesis?
No, there were no funding sources looking at hormesis at that stage of the game. It was kind of just a quirky dink.
What were the projects that you were working on at that time before you made this phone call?
Oh, I was working on various aspects of both toxicology and I was working with a group of our epidemiologists. We had a variety of projects. I was looking at, on the toxicology side, groups of individuals who had a predisposition for developing toxicity due to an enzyme deficiency. And I had animal models that allowed me to do that. I was working with epidemiologists, taking a look at high levels of sodium in drinking water in the greater Boston area on blood pressure in kids who are in school. And we found that there was a relationship, and that led to some very large funding by the US EPA for our work. I also got involved with another thing with children, which was really another environmental concern. The audience may remember Love Canal and Times Beach, Missouri, these major pollution episodes that got great national attention, and for a variety of reasons. But the one in Times Beach, Missouri, was tied into this chemical called dioxin, and it was tied into exposure to Agent Orange, the residual of Agent Orange that was within the Vietnam War. and it was this pesticide called 2,4,5-T. And when you manufactured it, dioxin would happen as an unintended byproduct. And dioxin was claimed to be the most powerful carcinogen on the planet at that particular point in time. And so, as it turns out, Times Beach, Missouri had massive contamination throughout the town of dioxin because of the illegal disposal of chemical waste from a facility. Put it that way.
I think part of the original story about radiation for cancer treatments comes from Agent Orange in World War II. Are you familiar with this? Where there was a ship that we sent over that had lots of Agent Orange in it, but it wasn't supposed to.
Oh, that was over in Italy when the ship exploded.
The Germans blew up the ship.
Yes.
And it radiated. I mean, like, it had this effect on all these people, and the people went to the hospital and they actually saw a benefit to it. And so it was adopted in to become what we know of as radiation therapy for cancer. But it started with this kind of horrible incident.
Yeah.
And that's really interesting. I mean, that's a great story.
Yeah, I think it affected like the folic acid metabolism.
Yeah,
and exactly. It led to Sidney Farber Hospital and Sidney Farber, the physician, leading to the first treatments and cures of leukemia in children.
That's right. That's right. And it was all like, we weren't supposed to have chemical weapons, but we wanted to have them there just in case the Germans decided to use them on us. We'd have them available to use back. And then the Germans exploded the ship. I mean, it wasn't an accident, but they found out the hard way that we also had them. But I mean, I always think this is sort of interesting, and it sounds tangential, but it's actually not, because I think a lot of what I want to get to and what you've discovered with, sort of the response to radiation at different levels also goes to our understanding of mutations.
The interesting thing here is that there's another extremism when it comes to the environment, and it's different than the dose response, but it just reflects the mindset of like a regulatory agency such as EPA, where they tend to try to err on the side of safety, and they do it to such an extreme that it becomes ineffective, and it destroys their credibility as a scientific organization. And this was one of my first encounters with that, was really on this dioxin issue, because the company that had the deep pockets and was responsible for the cleanup of the problem at Times Beach, they had to deal with the cancer risk assessment process. And so dioxin had been shown to be a carcinogen in an animal study, but in doing the risk assessment, the EPA had to estimate how much dioxin little kids might get from ingesting soil. And so they made the assumption, this is an amazing assumption, they made an assumption with no data. It doesn't sound surprising to me now in retrospect, right? But with no data, that the average kid would eat 10,000 milligrams or 10 grams of soil each day. It's a massive amount. And so they did the risk assessment at Times Beach, Missouri, under the assumption that the average kid would eat 10 grams a day. And so that led to there being the assumption there was massive risk of children developing cancer because they would sit there and just eat soil all day long, massive amounts. And so basically what that meant was that you had to clean up the soil to an impeccable degree to protect the children from doing that. Well, the scientists who were involved with the industry side said, that's a crazy assumption. There's no data to support that assumption. And so I was running some big meetings and doing some work on that area. And the industry group, they went to the CDC and the EPA, and they said, we need to have somebody do a study on how much soil kids eat. And they said, well, we don't know what you should do, but you should contact this guy Calabrese at the University of Massachusetts. And so I had, of course, never done a soil ingestion study, but who had anyways, right? And so they came to us and asked me to put together a study on it. And so our results were interesting because while the EPA was using 10,000 milligrams a day, our study came out that the average child was ingesting less than 50. There was a big difference here, and it was gigantic. And the implications were actually pretty gigantic in terms of the cost of cleanup, not just at Times Beach, but this was going to have an impact across the whole world. And so when the study was done, I was called into EPA in Washington to have a data sharing day with their exposure assessment group. They gave me the whole day. I brought the data set, let them see it, everything else involved. And so, as it turns out, the findings were sufficiently convincing to the EPA. Now, this is actually a major part of the story, that at that point in time, EPA actually could follow data compared to EPA today. Okay, and that is that when after I presented my data, within 6 weeks of that time, the U.S. EPA reduced their exposure estimate of 10,000 for the average child down to 200 for the upper 95th percentile of the population. And so they really accepted our work.
That's amazing. I feel like that's very hard to get them to come to the table. And
so
like
what did that mean to the company? Well, it saved them their liability at more than $5 billion on this cleanup. $5 billion back, that's like $1992, $1990. Today, I don't know what it would be worth, right? And so what happened, the interesting thing is that, wow, so we published it and we did everything we're supposed to do. But then the company, then because they were so appreciative that it saved the retirement plan from all their workers and everything, right? They sent me a personal check of $25,000 and said, thank you very much for your great study. And I said, oh God, I said, I can't accept this $25,000 check. I said, they would be like buying me out. I said, the compromise is that I'll give the check to UMass. They can spend it any which way they want to. It won't come to my research at all. Went on with the rest of my life. Well, don't do that sort of thing ever again, because what happened is that the company, reported that, to the IRS that they had given me a check for $25,000.
Oh.
That I didn't report it on my income, and the next thing I knew, I had a full audit from the IRS. So thank goodness that the University of Massachusetts is bureaucratic, and that the company was bureaucratic, and they could combine their
really went, yeah,
and they followed the flow of the money, and it didn't go to me. The real point here was that back in those days, the federal agencies and today, they have a very strong tendency to grossly overstate risks and to not do sufficient research to actually answer the question, and make an assumption. And the assumptions, in this case, and in many other cases, are shown to be totally wrong. And yet those assumptions drive cost, and they can have very devastating effects on the economy. And when it comes to the dose response, they can actually lead to preventing good things from happening at for certain agents at low doses. And they just get the whole thing wrong.
Let's switch gears a little bit and transition into what you've uncovered with the LNT model. And, sort of, if you can give us I don't want it to be brief, but I sort of concise history of, all the Nobel Prize winners and the sort of, the initial sort of research that didn't have any data, right? Let's just start there, and then we can kind of hop our way forward. Because I think the history of this is actually critical for people to understand, because we've been building upon a false foundation. So it's important to understand how the foundation was laid.
I'd have to say that while we're talking about dose response in great intensity in 2024, the actual original studies to begin to study what is the nature of the dose response, certainly in the low dose zone, was actually back in the 1880s. And it was done over in Germany. And it was really trying to come up with a new set of agents that could be used as antiseptic agents in sterile surgery, really, like by Joseph Lister of Listerine fame. And the person who was leading this charge was a young German professor by the name of Hugo Schultz. He was in Greifswald, Germany. And Schultz Schultz was just trying to find, some agents that could kill bacteria, and he wanted to see which ones could do it the best. But he used yeast as his little model, but he had mercury there. He had formaldehyde. He had lots of different toxic potential chemicals that he tested. And when he tested them with his yeast at high doses, he saw yes, they all actually killed the yeast. They slowed down its metabolism and killed them. But each of these agents at low doses, they stimulated the growth and metabolism of the yeast. And he thought that that's impossible. These are toxic agents. How could they be actually stimulating the survival, lifespan, and metabolism of the yeast? And so he thought he made a mistake, kind of like my professor did so many years later. And he does this study like me many, many, many times. And he kept seeing the same low dose stimulation, high dose inhibition. So he had something, but he made a terrible mistake, and this affected the course of biology up until today. Schultz talked with a professor who was very sympathetic to the medical practice of homeopathy. Okay, and homeopathy back in the 1880s in this country and in Europe was very prominent and was an equal rival, maybe even stronger than what I'm going to say, what would be called today traditional medicine. Uh, and traditional medicine back in the mid 1800s was very unlike what we have today. It was, it was more heroic medicine. I mean, anesthetics were just being discovered. I mean, how you did surgery for breast cancer, how you didn't have any medications to treat arthritis. You had nothing. Medicine back in the days of Gunsmoke on television with Doc Adams wasn't very sophisticated. Okay, not at all. And in fact, it was pretty scary when they're, trying to pull a bullet out of, Matt Dillon's shoulder with,
whiskey and a stick, right? You bite on the stick.
Exactly. It was lucky that we're born today rather than back in those days, right? Having babies and everything else was a little bit different, for your great, 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 great grandmother and so forth and so on, right? And so homeopathy was a much gentler way with a very vanishingly low doses of trying to treat people. And even if it didn't work, it might be working by a placebo effect. And that was better than being, heroically treated by Doc Adams with how they were working back in those days. So as it turns out, Hugo Schultz and this gentleman, they believed that they had discovered the explanatory principle of homeopathy. And there are some details behind it that I won't share with you at the present time. But what happened was that Schultz was in a traditional medical school. And it was like, so he's in a traditional medical school and he's telling his faculty and others that he's discovered the explanatory principle of the opponent of what they stand for. And that's like, it's kind of like being at Boston in Fenway Park and wearing a Yankees hat, right? You just don't want to do that. So
good luck.
So yeah. He's in this medical school and he's proclaiming that he's figured out homeopathy and it's right and it's the way things should go. Well, guess what happened to Schultz's career? It went south very quickly, and it became ridiculed. But it was interesting. Schultz and his followers within homeopathy, they developed the concept of dose response before traditional medicine did. Traditional medicine really hadn't worked out a dose response. So when traditional medicine figured out that they had to come up with an alternative, they could not accept the alternative of what? Their opponent. In fact, their enemy.
Was it just a political divide in that sense, or was it more? I mean, because I think like on some, when you look at these things in a larger context, you often find that the competitor or the political rival or the other association comes up with something that internally people recognize, well, that is working better than our thing. And it just takes a little while for them to find a way to adopt it and claim that they came up with it on their own, right? So,
well, they never went that direction. It was largely economic. It was who was going to control money and resources.
Yeah.
And as it turns out, they had to come up with their own dose response, and eventually they settled on the threshold dose response model. The threshold kind of, you know, it made common sense. Most people, like the alcohol example I gave earlier, alcohol suggests that there's a threshold. Lots of things suggest there's a threshold. And so it's almost as though they didn't have to work very hard to establish an alternative dose response model, which was the threshold. And so what happened is that, from in this country in 1900, there were around 23 homeopathic medical schools. And by 1920 or so, there were about three. Yeah, everything. It was like traditional medicine was really on an ascent. Homeopathy was really on a big descent, and Hugo Schultz's career was going nowhere and nowhere fast. And so what happened is that this biphasic dose response relationship that he showed, which will eventually in 1943 be given the name Hormesis by some U.S. scientists out at the University of Idaho, basically it developed a major opponent, and the opponent of the biphasic dose response, which will be hormesis, is going to become the biomedical community, what becomes traditional medicine. So pharmacology, toxicology, and medicine in general hated or disliked or opposing homeopathy and anything that was a component of that, and that was the biphasic dose response that will ultimately become hormesis. And so the entire 20th century, you have our really very brainy biomedical community developing with a belief that the basic dose response is what? A threshold, and that if you see a biphasic dose response, then what that is that it's just background noise. It's
attributable to something else.
Yeah, and that's at best. And so it was ridiculed, scorned, not funded, not taught, and was why I was not taught, and why people today still don't teach it. And so what happens is that I had to confront a reality that the people in power, the people who fund, the people who write the textbooks, they didn't give you any standing at all. This is telling you, the burdens that hormesis we're dealing with, but we're focused on the linear dose response. And this is a problem for the linear dose response too, because it wasn't on the playing field. Just like hormesis was pushed aside, threshold was the only player. But then this guy Herman Muller came around in the early 1920s, and he became a promoter of an alternative dose response for radiation-induced mutation. And he said that it was a linear dose response. And that linear dose response was, as I have described it before, there's no threshold. And that and even a single ionization can start this process of mutation, and that can ultimately lead to, as the mechanism of cancer, and ultimately the development of all sorts of human and animal cancers. And
so just so that everybody understands this, I feel like we should explain this again, that the linear dose response essentially means that there's a consistent ratio that goes down in a consistent fashion that never leads to no harm, never leads to a positive outcome, but it's just a sort of like a risk that goes up as the dose goes up and goes down as the dose goes down, but is harmful the whole path.
Exactly. That's a good way to say it. And that is, but back in Muller's era. Now, Muller became a graduate student in 1911 at Columbia University. And people were just beginning to, they weren't interested in risk assessment, cancer risk assessment back in 110 years ago. They were actually the big questions back in those days were trying to figure out what was the mechanism of biological evolution. There was the origin of the species, Darwin's Origin of the Species. There was Gregor Mendel in terms of trying to come up with understanding genes and sexual reproduction and how they distributed in populations. But that didn't explain the mechanism of evolution. They knew about the gene concept, but how did we get here? What creates biological novelty? How does life actually work, and what's the evolutionary mechanism? And so Muller was studying with his professor at Columbia fruit flies.
And fruit flies are commonly studied, probably important to mention, because they reproduce a lot. So if you're trying to look at genetic changes, they can be sort of easy to control. I mean, theoretically, easy to control. The control groups should look similar, which is something we'll hear more about. And that they, because of the quick reproduction, it's interesting to look at their genetic changes over time.
Yeah, you can get 10 to 12 generations a year. You work with plants, you get one. So you can it's a big difference, and that's going to play a role here too. And so what happened is that Muller and his group, they had to figure out how nature induced mutations in fruit flies and other organisms to try to figure out evolution. So they had this little creature, and they knew that it could induce mutations because they had seen they could see visual mutations. Normally, red eyes. Sometimes you would see infrequently, but you would see some offspring that had white eyes. You knew that there was a mutation there, and then there could be other sorts of visual mutations. But the interesting, they threw everything except the kitchen sink at their little fruit fly. The most toxic chemicals you could give. They exposed them to X-rays, to radium. They did everything you could do to induce. They made the temperature hot. They made the temperature cold. They did whatever you could torture your little fruit flies to the extent that you could possibly do this, right? And they could not induce gene mutation in these fruit flies. This. You might think, well, I mean, really, they couldn't do that. What was their problem? I mean, they weren't that smart if they couldn't figure that out, right? But they couldn't figure it out, and they were brainy people. They really were. And so, as it turns out, what was the problem for Muller and others is that Muller and others have stated that they had observed about 25 million fruit flies, and they claimed of these 25 million fruit flies, just picture that, right? That they had observed 400 visual mutations. That's very infrequent, one in about 50,000. I mean, if you were just nodding off a little bit in the lab, you could miss one or miss two, and then you have to wait another 100,000 animals to see another one. Very, very infrequent. And so it was.
By like any scale, that would be a failure to replicate, right? Like there's nothing here. We see, like statistically, this is a nothing.
Exactly.
Move on. Right.
The only way that evolution can actually happen is through mutation, but mutation is so rare that how does it happen? Muller makes a major mistake here, and this is going to determine the whole course of risk assessment in EPA that we experience today. This is the fundamental mistake. This is the original sin of risk assessment. Okay, we can make this very theological, and that is that. So, seeing what he observed, this 400 mutations out of 25 million, Muller concludes, I know evolution's real, but how is it going to happen? So Muller concludes that, well, given that mutation occurs, but is so darn infrequent, then the only way that we can have novelty preserved is that whatever mutations occur, and most will be negative, but some will be positive, then the body cannot repair any damage. So we have to preserve all the mutations we have, and that natural selection would get rid of the lousy ones and retain the good ones. And so Muller made an assumption back in actually the late 19 teens that in fact repair of genetic damage did not occur, because if it occurred, we really wouldn't have enough novelty built into the system to account for evolutionary change.
And I think this is so important that I just want us to like pause for a second here, because we know this now is not true, right? Like we've done enough research, we've developed tools that can look more closely. We know cells repair, we know cells, deconstruct or self-destruct, right? And are swept away, and they're all, through apoptosis and all of these other things. We know that this is factually inaccurate, but it became the presiding presumption because of his work that then now downstream we are still seeing consequences for. So when you call it the original sin, it's the original sin for EPA. It's also the original sin for a lot of cancer understanding of cancer mutation, and this notion of, I mean, what I would say ultimately is sort of like some homeostasis that we have in the body, which is that the body is excellent at dealing with damage and repair. And that part of the aging process is that that slows down, right? Or it may not be as effective. But that certainly this notion that a cell can repair itself is something that we can say fairly conclusively. I mean, there's no absolute certainty, but that seems to be something that we've accepted today, that he challenged and said was wrong, or he didn't even really look at. I mean, did he consider that hypothesis, or was he just so hell-bent on what he was predetermined to believe that he didn't consider an alternative?
Well, you raised really the fundamental question, and that was that Muller didn't propose any alternative hypothesis or a way of testing between hypotheses. Muller had a valid observation of 400 visual mutations out of, let's say, 25 million fruit flies. Valid observation. Muller concludes that, well, how did that come about? Well, it comes about because, the genome is very stable. The genome is extremely stable. Well, you could come out, as I'm sitting here, you could come up with the exact same observations by saying the exact opposite. The genome is very unstable, but you have a phenomenal repair capacity, and you end up with 400 mutations out of 25 million. You could explain that very easily by his hypothesis or my hypothesis.
So if we even go back to your early experience, if Muller had had your professor with the peppermint plants, the professor would have said to him, like, okay, just as he had you go and remove the soil and isolate the variables, somebody could have said that. I mean, if at Columbia, Muller's, advisor had said, okay, what's the alternative? We might not be in the place that we are today. It wasn't really even seriously considered.
Exactly. And so Muller built into his thinking, his thought process, that in fact repair doesn't exist. Now, what does that actually mean in terms of LNT today? Well, see, what happens is that Muller had that thought process when he was a graduate student, and after he got his PhD, and now he gets his PhD in, I think, 1915, but in 1927, he publishes his major paper in Science, in which he shows that high doses of radiation can induce gene mutation.
And I would say he doesn't show it because he has no data for it, right? But he claims.
That's right. He claimed that in his science paper. So he's now found, for the very first time in his mind, he's now found a cause of mutation. Up until that point, now it's been like 1911 to 1926 to 27. It's been 16 years. Nobody's found any cause of mutation, despite all the work that has been going on this. So he's now become, you might say, it's not quite true, but he's become, let's say, the first to claim that he has induced gene mutation, and by some means, and he's using high doses of X-rays. Now, as it turns out, Muller actually is giving X-rays at a dose rate that is about 100 million times what I'm experiencing right now, background radiation. 100 million times, not like 100 times more, or 1,000 times more, or 1,000,000 times more. It's 100 million times higher mutation rate.
If we put that into the human realm, that would be like having a nuclear bomb go outside my door, right? I mean,
well, in very flowery language, put it that way. But the idea is that just shows you how low background radiation actually is. That we're being exposed from cosmic rays and background radiation. And so Muller, what he does is, for him to explain evolution, he has to say, oh my goodness, what is the cause of evolution? I think it's background radiation. But the only way that he can explain that is that he has to say that the relationship is a linear dose response all the way to a single ionization, and it applies.
Which he hasn't tested. He's very far on the other end of the spectrum in terms of his experiment.
Exactly right. From that, you might say, vague and unsubstantiated origin, Muller comes up with the explanation for evolution in a 1929 paper, and he eventually shows that the background radiation, which was the cause of evolution, um, it's kind of murky for him. But he has the only way that he can account for it is in this linear fashion. But he knew, based on a study that he did with a physicist in 1930, that background radiation, even though according to Muller it was a mutagen, it could not explain even 1/1300th of the amount of mutations that in fact occurred in his control group. The very, very background radiation, even assuming a linear dose response, has almost no impact on your genome. Very significant. But what this whole thing led to is when Muller came up with, after his paper, he's now getting into the linear dose response.
I think it's also really important because we talk a lot at Broken Science and, sort of generally about sort of corrupted means that science gets perpetuated. And in this foundational paper, which ends up being the sort of bedrock for him going on and winning a Nobel Prize and having this storied career, there is no data shared. You can't find any data, correct?
In the science paper, that's right.
And his advisor at Columbia is very close with the editor of Science magazine. So there's a lot of, we don't want to say anything conclusive about it, but this corruption we see and we talk a lot about today in terms of revolving door and funding sources and all of that was very much alive and well at this time, 100 years ago, that sort of helped to push forward his research without there being real methods of validating what his claims were.
Well, that's exactly right. But the interesting thing is that Muller has his moment in the fall of 26 and tries to publish his paper in 1927, and he somehow, on the most important issue of the day, which is basically this evolutionary question, and can you induce gene mutation, the biggest question of all, and the biggest journal in the world, which was Science, he publishes his paper and he only discusses his data, never shows his data. And this drove everybody crazy. Could Muller do this? Muller had made a couple mistakes in the past, and his advisor thought that he really destroyed his career at this point in time. Did he have the data? Did he have the goods? And why would Cattell? Muller wasn't a big-time scientist at the time. He was kind of like the equivalent of I'm just an average professor at the time, right? And nothing significant really emerging from him. And he let him in there. He gave him a great advantage. He gave him a great advantage over the competition who was trying to come up with this as well.
And we're not far behind, right? I mean, it was
3 months behind. The closest were 3 months behind. And so Muller then goes over to Germany and he shows his data. He actually shows his data. And what he showed was convincing to the audience that he had induced gene mutation. He became a superstar overnight. He publishes his paper. This is very interesting. He publishes it.
That was at a conference. I think it's also important to mention that wasn't peer reviewed. It wasn't in a journal. It wasn't scrutinized by anybody. It was a presentation he gave.
Yeah, it was a presentation, and it was a poorly written paper. It didn't have methods and materials. It didn't cite any other person's work. It was sloppily written. And Muller was quite aware of the peer review process. His advisor helped create it in different formats and frameworks, and Muller was definitely fully aware of it because I've gone back into the history of peer review and had to track all that down. And the interesting thing here, too, one of my little discoveries is that I found, because I always wondered, well, how did this deal get worked out? I mean, I would love to get a paper in Science with no data. I've tried many times with data and I can't get into Science. It's a tough road to hoe to get into that journal, right? And so, but Muller gets in with nothing. And so what I learned was that Muller and the editor and owner at that point of Science, this Cattell guy, Cattell worked a deal out with Muller, and I have it in writing, that he owned two journals. He owned Science and he owned something called the American Naturalist. He told Muller that he wanted to have a special issue of the American Naturalist, and he wanted Muller to select papers that were excellent, that were presented at the conference over in Berlin, and that Muller could invite these people, and Muller would be the sole determinant of what went into that special issue of the American Naturalist. And that's an awful lot of work, especially in snail mail and everything else, peer review, getting all these things together. That could just consume you for 6 months, 8 months. I mean, it would be that.
It wasn't a paid position, though. It sounds like it was kind of a quid pro quo, right?
It was not, from what I can see, it was definitely not paid, and several things actually happened with this that are very weird. So Muller goes forth and does this, and
I feel like, just from a strategic perspective, it gives Muller a lot of power over his competition, not just because he's getting his paper, accepted and whatever, but he also is gatekeeping who can publish in this other, which was also a very prominent journal. It wasn't like a fly-by-night project. It was a very powerful journal at the time, right?
And it still is. It's still a powerful journal today.
So Muller, that's a win-win for Muller, really. I mean, it's a lot of work, but he's getting to be essentially the editor of this other publication by accepting or denying who gets to publish there, as well as getting to put his own work front and center. I mean, it's a huge conflict of interest by, any sort of moral, intellectual integrity standards.
I would have to tell you that Muller's chief competition also presented at that same conference. And so Muller would have had the capacity to have highlighted their work, but he didn't.
Shocking, right?
But the other thing is that Muller could have presented his paper that was non-peer reviewed in that publication and put it through. He could have put his own work through a peer review. This is a really interesting point. Muller chose not to put his own paper in the American Naturalist with data. He could have even had it peer reviewed. Muller, I'm sure, made a conscious decision to keep his paper in a conference proceeding that almost nobody could find or read, very poorly cited. And that was where the sloppy paper, and all the attention will be given to Science Journal, where Muller says he induced the artificial transmutation of the gene, and that's where his Nobel Prize will come from. And so Muller had it set up right from the beginning to do this. And a couple of things are interesting. This is a big exciting moment in science with what he's claiming. So Muller's department chair, John Patterson, he wanted to extend Muller's work to somatic mutations, which are Muller worked with reproductive cell mutations. So he looks at exposing fertilized eggs and young larvae to high doses of X-rays. And he wants to see if it changes the eye color from red to white. And he's got ways to do this. And as it turns out, he published a whole series of very large studies on this. He couldn't induce gene mutation, no matter what he did within this setting. It was, if anything, he had a threshold, but he had a very difficult time in inducing gene mutation. Put it that way. There was nothing that supported an LNT in this Patterson's work. And George Snell wanted to extend Muller's fruit fly work to a mouse model because, humans are a mammalian system. And so Muller thought that was a great idea, invited him down and said, in fact, I was going to do that myself. I have 400 mouse cages. I have strains of mice. Yeah, come down and help me do that work. We'll give you the lead on that. You can do it for your postdoc with me. And so the guy goes down to work with Muller, and he does under Muller's guidance, he does a very good study. And guess what? He cannot induce gene mutation with very high doses. So what happens? This should be a very big story, right? It gets swept under the rug. The guy goes down to see Muller because he's so inspired by Muller's paper in Science. When he writes his paper, he never cites Muller's work at all, ever. It's like he shares. It's like it's really swept under the rug. Muller, in his long career after that, never once cites this guy's work. It was hidden. And what's the social climate down there? Because Muller's now famous because he induced gene mutation. What's it going to mean for humans? And this guy can't show that it happened. Well, this guy leaves Texas, goes up to Jackson Labs up in Maine, which is a big mouse research center, and he eventually himself wins the Nobel Prize in 1980 for immunogenetics. He was a superstar on his own. And he always kept his mouth shut.
That's a really important just biological point to make, that the effects that Muller was seeing at these high doses weren't necessarily mutations in the way that we think of them, but they were chromosomal changes that were coming through and may have appeared as though they were mutations, right?
That's what ultimately has been learned, that Muller deluded himself. In fact, I'd have to say that deluded himself is probably not right. Muller wanted the prize so badly that he tried to perhaps convince himself and certainly convince the world that his incorrect interpretation was the right interpretation, that he had induced.
It's a cognitive dissonance sort of 101, right? Like just help that on showing that you are right in some capacity, even despite all the evidence. And I think, when we talk about scientific integrity, another thing that comes up here is this notion that like somebody goes and they try to replicate your results, right, in a slightly different environment because it's a mammalian model rather than the fruit flies. And they're not able to. And instead of saying, wow, that's really interesting, I wonder if this is because of the difference in the animal or any of these other things, it is ignored, deleted. And the person who had those findings agrees for their own sort of political survival to not say, wait a minute, I found something different. What you're saying can't be conclusively the way you said it. This is really, it should be, in my naive brain, it's a really exciting discovery because falsifying something should be just as exciting as finding something, right? And essentially what he did was he falsified Muller's earlier work. But instead of that being accepted into the literature and the community, it's silenced, rejected, put in a desk drawer, close the desk drawer, you can keep moving on. And sure enough, he is rewarded. I mean, it sounds like his work is warranted of his reward, but he wins the Nobel Prize later on. Now, if he had spoken up, we can't say what would have happened, but it's given how powerful Muller was at this stage in history, it's unlikely that he would have been put forward in the scientific community as much. Now, this is important for everybody because this is really the opposite of what you want in terms of science, the evolution of ideas, right? You want somebody to sound the alarm and say, I couldn't replicate what you did. And I don't know why, but we need to go back to this foundational work and understand it better because it's not translating the way that you have assumed or inferred or demanded it does. It doesn't have the impact or the outcome that you're claiming it does. And that did not happen at this interjection. And it doesn't happen again later. But for this is the first time that somebody has really said, wait a minute, this can't be quite what you're claiming it to be. And it goes nowhere until you brought it all to light.
Well, it's so interesting because I work closely with some people who are extremely well-trained careers in the world of mammalian genetics, radiation genetics, and they never heard of this work until I brought it to them. It was, and their advisors were very knowledgeable of Muller and these other groups. Worked with this guy up at Jackson Labs and still didn't know what he did when he was with Muller. It was like he kept it, like he didn't want to get the raise Muller's anger. Muller was a very tough guy and could be very harsh and penalizing and attacking and could destroy the career of and probably anybody in a young person. You had to be very, very strong to counteract him. And I've written a lot about that.
So I want to just ask you on sort of in terms of your story, how did you come across this? I know you've purchased a ton of correspondence of Muller's, but where were you when you found this guy's research and you started to process?
The Schnell's work or Muller's original problem?
Schnell's, like this was
God. The Snell's was within the last 9 months. What happened is that you have intuitions, and I read widely, and you see something that's a little bit a little bit different, and you might just pass over it because you're moving on. But it's that little light beam that goes by that well, maybe that was just a little flicker of something else. But no, I'm going to follow that lead. And so it was a very subtle thing that because I didn't really know about George Snell, and then I began to read about him, and then I got more curious and curious and curious, and then it led me into. Thank goodness he was a Nobel Prize winner, because I got letters between him and Muller. I got letters between him and some other people. I then dove into everything that was about this fellow George Snell, and and then was able to re, able to reconstruct what happened to the extent I think possible today. And it's just too bad that that I didn't know this, so many years ago, so one could have interviewed him while he was alive, and really, because he held this close to the vest. Muller was a godlike creature, and it's very difficult to challenge that sort of person. And so even though this guy Snell was a Nobel Prize winner, he just he kept the lanes parallel. He would not intersect with. He could have criticized Muller, made it very public. He could have, after Muller was dead, he could have done that, but he didn't want to bring probably the harsh criticism from the Muller sycophants on him. And so he kept his life very pure and nice and straight line, and it's like going to a faculty meeting at any university, and you know what the political correctness code is, and then you just kind of nod your head and make the person in charge think that you agree with them, because if you disagree in public, then you can pay a price for that in one way or another, and that's true in so many different walks of life. That how can you bring out honest reactions when you really don't want honest reactions? You just want to control people, and I see that in all walks of life, actually. And so, and to challenge it really makes you a target. It makes you a target. And Snell, I think Snell would have affected social relationships, professional advancement, invitations. So he just kept, friendly with everybody, kept it quiet, did the best he could possibly do to himself, even sweep it under the rug. And
so we're going through the correspondence. I assume this is like from the Nobel archive.
These actually came from the Jackson Lab archives.
Okay. And so what was that moment like for you? I mean, you had this intuition or instinct that there was something more to this, right? That somebody had to have looked at this and had the same inclination that you now have had much later. But to find that there was a contemporary that actually had done the work, had refuted the claim, had kept it quiet. And this is something that you've been looking at. I mean, I think of you as an investigative reporter with the amount of work that you have done and the digging over time to sort of put these pieces together. So I know personally what that's like when you find something and you're just like, oh my God, like how this is it, right? This is
I've had a whole series of these wow moments in this. And that was one of them.
Were you nervous to share that with colleagues in the radiation world?
I'm aggressive in my wanting to share it.
Okay.
And I move it as fast as I possibly can. I always play the game the way I think you're supposed to. I mean, I go to the top journals in my field. I submitted the Snell paper to this is very contemporary, right? Very contemporary. I submitted this Snell paper that is very unique. I wrote it from scratch. Okay. Everything is cited exactly as it should be. I sent it to a leading publication, and within 24 hours, it was sent back to me rejected by the editor-in-chief, saying that I had failed their plagiarism test. Okay.
You're like, wait, I've already attributed this to snail. How is it plagiarism?
And so what I did was I have a colleague who is an associate editor at a major journal. I mean, and I have the software myself, but he has it. And I said I wanted an independent person to do a plagiarism test with the same software that they do with this journal. And so I run it. I said I can't imagine I can't imagine I would fail. So he ran this my manuscript, and he said Ed, he says, you didn't fail the similarity index, however you want to view this. You were way under it. And in fact, he said, I know the cutoff point for that journal, and you were way under the similarity for that journal. So I write back to the editor in chief and I say, well, thank you very much for telling me I failed the plagiarism test. I don't really, I'm not an expert, but we'll work this thing out and try to resolve it, because I don't want this to happen again with your journal or another journal. I want to solve this. Now it's been about 2 months, and the person doesn't respond to my requests. I've written probably 3 times. So I then sent it to an alternative journal. Okay. And the alternative journal, which is a very high level journal, it went through their peer review, and it has been accepted. And it's now just been recently posted on PubMed, and it's there for the world to see. And so,
That's great.
But nonetheless, I'm not letting this editor off the hook, because he has to be, or she, without giving it away, has to be accountable for what they said to me. And I want to learn. I suspect that I was put through the plagiarism review, but they all do it these days. And I passed that other journal, right? So I'm trying to see what's happening. But this is an example of what I have to go through in a lot of circumstances, because my work has offended the status quo in a lot of different ways. And I can tell you that I have been shut down from probably 7 or 8 journals to submit papers on this topic, just because I have offended the National Academy of Sciences, I've offended the Journal of Science, I've offended Nobel Prize winners, I've offended the EPA, and they all have friends in high places, and have ways to squeeze you in ways that I can feel, that may be hidden, in all these various subterfuge ways that people in power, hide how they get at you. But and so I've had to fight this fight for a long time.
Okay, so let's get back to the story.
Right.
Your story is just as interesting to me, frankly, because you've stayed with this.
Yes.
And your intuition has been right, and in the sense of you've had a hunch about something, you've tracked down data or correspondence that have corroborated and, sort of in a fact-checking kind of way, allowed you to then build other threads to connect all these other pieces. So
I'm going to give you the connection that's very significant. There are 2 things. One, when Muller did come back from Berlin, and he was acclaimed by the world that he made this great gene mutation discovery. His best friend, Edgar Altenberg, he goes and he says to Muller, he says, Herman, how do you know that you didn't just knock big holes in the chromosomes? How do you know? And Muller didn't know, even though he claimed that he couldn't. He never did. He was just these tiny point mutations. And so Muller knew that he was ultimately going to have to face the music at some point in time. And Muller then...
That's private correspondence that you've acquired that he never publicly acknowledged that.
No, he publicly brought out, when he gave a talk at the National Academy of Sciences, he brought out that his best friend made this assertion as soon as he came back from Berlin. And so, and I have evidence. I do have evidence that Muller came up with a way to try to test it, and he got a person on sabbatical from Washington University to try to test this hypothesis that ultimately, failed repeatedly over time. But despite Muller's failings, the real key that was going to be happening to Muller was that there was this woman who was a Nobel Prize winner herself, Barbara McClintock, and she was a plant cytogeneticist. And so she had an advanced way of of seeing the great resolution on of chromosomes under the microscope. And so, as it turns out, because she was very advanced, she was offered a job at the University of Missouri to work with this guy Louis Stadler. Stadler was a rival of Muller, was really only 3 months behind Muller. And ultimately, he was only 3 months behind Muller. But after they published their papers, Muller won the battle by 3 months. It's going to get all the publicity. But Stadler invited her for a faculty position at Missouri. She went there, and Stadler's showing her his research with corn, with showing mutations with the bacteria, with the radiation, and she looks at his work with her new cytogenetic techniques, and she said, "You think you've induced gene mutation?" He says, "Absolutely." He says, "You just knocked big holes in those chromosomes." And he got angry and upset. He argued and debated with her. After a couple of months, he ended up having to agree with her. She held her ground, and he eventually became educated. And so when he got over feeling, sorry for himself, he then began to say, well, if I'm wrong, then guess who else must be wrong? And he says, I think that Muller fell victim to the same flaw that I fell through. And so Stadler then became the biggest and profound challenger to Muller, because he concluded that Muller confused an observation with a mechanism. But the interesting thing is with Muller is that he went to Germany in 1932-33, and he got involved with some very heavy-duty physicists and people who are radiation geneticists as well. And together, they put together this linear dose response model that Muller was thinking about, but they did it with a mechanism. And their mechanism was the mechanism of target theory from physics. And so, but built into their model, and this is the whole key for EPA and where we are today. Because of Muller's flawed evolutionary explanation that we've talked about, they decided not to include a repair component in their linear dose response model. So they made it so that every single hit had an effect. No hit and effect could ever be repaired. And that became the basis of the LNT single hit model that was done and published in 1935. 40 years later, they gave it to US EPA, unaltered, you might say, and that's what was used to guide risk assessment for cancer, for chemicals and radiation in this country. And because of our dominance, the rest of the world followed it. And it all started with that original, what I call the original sin, that led to the exclusion of the repair component, that ultimately led to, and repair was eventually discovered and so forth all along the way. And so there's a lot of subterfuge that happens between then and the present time, but as a twig is bent, so grows the tree, and Muller had a long reach, an awful long reach, but Muller started it all, and it started innocently with his mistake about evolution, and then he transformed it into will ultimately be hereditary and cancer risk assessment mistakes.
So when you look at this from the sort of larger perspective of things that we're doing today in medicine that are, let's say, based on or connected to these earlier flawed theses, what do you see as the biggest output in that regard? Is it cancer risk? Is it the way that we think about toxins at low doses? One of the other things that came up in the research that I did that I thought was really interesting was the panel that had a couple of people looking at bacteria and actual human radiation exposure, going over to Japan. The US government and the Japanese government worked together on these things, and how that was disregarded, getting into the Rockefeller Foundation and all that funding. That seems like another iteration of this story where things could have been course corrected and they weren't, right? And that the research that was kind of counter-indicating of this earlier work was literally ignored, right? There were 3 people on that panel who decided not to be on the panel. You should explain that because you're obviously more knowledgeable than I am about it. But I feel like that's another iteration of the story that's really important to explain how we got to where we are today.
You might think that Mueller is the big bad guy in this, and he really is. But Mueller was not alone. It was a culture that Mueller helped to create. There was a certain mindset, a certain way of thinking, and you couldn't think differently. If you did, you were excluded from the group, and that's really why George Snell slithered away and never said anything, because he didn't really want to experience, professional attacks and exclusions and so forth. They wanted to be part of the group, or his success was not going to be that great. And so when you go forward, you can see that this group of radiation geneticists, they held together pretty tightly, and they developed a certain belief system, and they wouldn't mind lying to advance their field and their careers. I have it in writing where they said it's okay to stretch a point because we have to actually frighten the American public so we can get more grant money. I mean, I've published papers on that by people who are on these National Academy of Sciences committees, right? I mean, these are the people we trust, right? I have lies from the president of the National Academy of Sciences back at the time of 1956, when the first National Academy report came out recommending LNT, lying to the New York Times, lying to the Washington Post, lying to whomever listened to them to promote the agenda that they wanted. The former director of the U.S. National Cancer Institute, Arthur Upton, right? Arthur Upton and William Russell did a big study on radiation and longevity in cancer, and they found no treatment effect, and they refused to publish it. They hid it. They published it 35 years later, so Upton could win a court case over in England for the defense, and discovered this story and revealed it. You have so many different stories of subterfuge. Self-interest is a very big deal here. It's a very big deal of scientists trying to get grant money. People think that the only bad guys in the world, money-wise, are those from industry and industry consultants. I'll tell you, academics go hand in hand with these with self-interest. It is self-interest. However you find self-interest, you have to be cautious and skeptical of everyone and what their self-interest is, and so.
I mean, I feel like that tenure is supposed to have some protection against that, right? But it doesn't seem to be effective. I mean,
it's all about money. It's all about your next grant, and that's really when you're in academia. Tenure, yeah, okay. I got tenure in 1980. It's 44 years later, right? You're only as good as your next grant. Okay, it's like a baseball player. Yeah, I was a Triple Crown winner last year in the American League. Yeah, but you're batting only 200 right now. What good are you, right? It's how good are you? Because it affects your reputation, your invitations. It's the swirl around you.
So then, why have you taken this on, given all that risk?
I think I have a problem with you might say it's a search for truth, but I probably have a problem with authority that I haven't resolved. I have to go back into, you really need to have a clinical psychiatrist here at the same time, going back into my relationship with my father and whether I resolved, my authority complexes. I've had problems in every single job that I've had. I have authority issues that I think I've been always right on, and then I think the authority's been wrong on. But I've got this stubborn streak in me, and it has cost me jobs. And although every job I've lost, I'd have to say that I've landed well. I'll put it that way. And so I could offend plaintiff and defendant easily because I don't really care what they think.
I mean, that is the theme in this show, right? Is that all of these people, and I've said in other interviews that I feel like I suffer from this too, and I think it's a genetic defect because we probably all would have been killed 100 years ago. but it's also the only source for progress. So standing up and putting yourself on the line and saying, hey, this isn't quite how it seems. And I've worked really hard. This isn't just being rebellious. It's you have done a ton of work to look into how this went wrong and how it was maligned in the political world, right? And how people were silenced. And what it has led to is a false sense of security around some scientific concepts that are hugely, hugely important today. I mean, like at the heart of some of this is even this notion of like genetic cancers.
I can tell you for sure that when I was interviewed in this 22 episode documentary by the Health Physics Society that I hope that you've seen, the person who was putting it together, the president of the Health Physics Society, John Cardarelli, after the fact, I'll tell you, we went through every single episode, and he absolutely demanded, I need to see your original documentation. I need to see that letter from Mueller that you claimed. I need to see this. If you can't produce that letter, this isn't going in. If you can't produce that, that isn't going in. If your credibility is on the line, this boom, 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 boom. It was the best thing that ever happened to me because I could produce every single thing that they demanded. It wasn't like I was telling a story that wasn't accurate. And when they got the letters, how I characterized them was accurate. This is all part of this process of this publication process. It's where people who are intelligent and experienced, and they're saying, I've never seen this before, and I'm not going to believe you unless you show me what you have in your hands.
Which is exactly what you would do, right? I mean, this is like what we wish that people had done with Mueller. Like this is the
exactly
you need
right.
to verify the information that you're claiming, and I mean, it's fact checking, it's validation in a scientific experiment, it's replication, it's looking at the same thing and figuring out, is there another way to interpret this? That's all we want. All of that. That's what's been missing from the history that's led to the problem.
Yeah, and, so I say, what's the problem with that? This is the fun part of it, right? This is
right.
It's my only regret is that Mueller still isn't alive.
Yeah.
And I can tell you that I have requested to get things in the Mueller files from the library, and there are certain papers that are so sensitive, apparently, that they will not share with me, and that it's controlled by the quote unquote family, and the family will not let me see some of the letters. And so Mueller's been dead over 50 years. I mean, give me a break, right? I mean, what happens after these people die? Is it going to be the great-great-grandsons and daughters? I mean, I have letters of an affair between Mueller and a girlfriend in Europe during the 1930s, right? But they were her papers, not his papers, because she was a prominent writer. So I have a bunch of love letters between Mueller.
How did you get those?
Oh, because I can smell things. I know how to find things, right? And so I haven't, for me, it just told me more about Mueller and his circumstance. It hasn't played part of my scientific story, but I know a lot more than I've written, and that's what I'm probably saying. But that would be a type of letter that the family might not let out. But the lady that Mueller sent his letters to, well, her letters were preserved, and there were no restrictions on those letters to me. So I got those letters, and so yeah, it's not this is the first time I've ever mentioned that I had those letters to anybody.
Well, that's great. I mean, I feel like that one of the tricks as a reporter is you can FOIA, so like Freedom of Information Act, right? Anybody who has a government email address or has taken money from the government, a lot of people don't know that. So if you can figure out who somebody's been corresponding with, you can go, and they won't give you anything, but you can go the other route, and the government has to get, I mean, they'll maybe try to put up a fight, but usually they don't care if it's some random person.
Along those same lines, and it goes with this story, and that is that about 4 years ago, I was invited to give a plenary address at the Health Physics Society on LNT. And so it's a big deal. They pay you away. You stay there for a week, get to meet everybody, have a great time, and schmooze, and then come back home, right? It's nice. And so 6 weeks later, I'm not sure this ever happened to you, but never happened to me. Now, I've had many invitations, right? 6 weeks later, I get disinvited by the person who invited me. And so I say, well, why was I why did you make it? Well, it was an administrative glitch. And I said, I'm too darn old. I've got too much experience. I know that you don't have an administrative glitch for a person who's giving a plenary address. It just doesn't happen. I have to speak to your boss, right? And then what happened is that I speak to the boss and they give the same false story. And then the third time up to the next boss, and the last boss in line says, don't call anybody else. You're not speaking here no matter what you do. It's all done for you. And so, as it turns out, several years later, I had the occasion of FOIAing a number of people. And as it turns out, these were government people, and you can do FOIAs on U.S. federal employees and some academics and the like. And I got a whole series of FOIAs. And guess what? And for a variety of reasons, I got the reformulation of how I got disinvited in this FOIA, and how they were so upset over my stance on LNT and how I would more or less pollute the audience with my views.
Wow.
That they came up with a story, this fabricated story of an administrative glitch. And so then and then I was replaced by one of the three people who made the decision. And so that person replaced me. So then last year I was invited to go back and kind of make amends, right? They invited me to go back and to now I could give a real, plenary address at the health physics conference, right? And so now you'll find this to be absolutely amusing, right? So what am I going to talk about at this meeting? So I say, well, I want to tell you a little bit about Herman Muller. Herman Muller in 1954 was targeted to give a big talk on his field at the first ever Atoms for Peace conference in Geneva that President Eisenhower was behind, big time behind. And so our Atomic Energy Commission really did not like Muller at all and his views on LNT. And they knew that he was going to spread corruption with his ideas if he moved to Geneva. So what they did was they got Muller disinvited. And he had taken his family to Europe. It was going to be a big deal. And then they said they pulled the rug out from under poor old Muller, right? And so this ultimately became exposed at the meeting, the whole thing. The New York Times, the Washington Post, the Journal of Science, and other groups, they all came to Muller's defense. It embarrassed the heck out of the Atomic Energy Commission. The Eisenhower administration was really upset with the director, the whole thing on this. It made him look terribly bad. Okay, so Muller became a hero instead of, a critic. And so now it's 60 years later, 70 years later, right? And so I go into this group and I tell them the story about Muller. I said, guess what? Do you think there's ever been another person who's been disinvited to the conference? And the guy said, does anybody know where that's happened? And nobody spoke up in this big audience. And I said, well, I know of one. And I said, and guess where it happened? I said, it happened in your society, the Health Physics Society. And guess who it happened to? It happened to me. And guess what? The people who made that decision are in the audience today.
I love it.
And I can tell you, after you can only imagine.
That's your swan song. You're never being invited back there again.
You can imagine that I'm loved and hated.
Yes. This sort of symmetry of experience and how one is demonized and the other one is like used as a model to replicate against you. And then they invite you back. I like it. That's a fantastic story.
And I told them, I said, guess what? Muller was ultimately, praised by the New York Times and the Post and Science. And a Nobel Prize winner wrote the editorial in Science about Muller saying how he was treated unfairly and all this. And I said, guess how many people came to my defense? I said, none. Okay, that's the big difference here. I said, when you were in favor of LNT and scaring everybody came to your defense, right? Because I was on the other side of the view. Who came to my defense? Nobody.
Where we are today, if all of your work is really exposed on a mass level, right? How does that change current guidelines or ways of thinking?
Well, that's a big question because my work actually goes beyond this historical thing, and I have other contemporary science things. For example, when when the LNT model was being rigorously looked at, within EPA back in the mid 70s and all, it had two basic assumptions. One of the basic assumptions, there are two. One was that all cancers arise in a single cell, and then there's clonal expansion of that cell, and then new tumors will come from that single cell. And then there's another assumption, and that's just the second two assumptions, and that is that there is such a thing as additive to background. So that what that means is that we have a background cancer incidence, and then when we have pollution from our environment, that it adds to an already ongoing process, and it makes it worse. And so that's the basic assumption.
Cumulative effect.
A cumulative effect, exactly. So what I did was I said, well, that's a basic assumption, and there's the assumption that they have to. This additive effect means they have to act by exactly the same mechanism. So that was put together and put forth in 1976. So 40 years later, I decided I'm going to go and test that hypothesis, right? Now, this is after oncogenes and big insights into the molecular basis of cancer. So I go and I try to take a look at, okay, what are the underlying genetic mechanisms of cancer for background tumors? And I take a look at 45 different carcinogens, including ionizing radiation, in 13 different animal models. And I find no support for that assumption. No, none, zero. And so I published a large paper on that basically totally challenges one of the fundamental assumptions. And I have other challenges that are very contemporary. And and that's why the Hormesis model becomes important here. I have an alternative model, and for dose response and carcinogenesis and so forth. So I'm this critic of the historical foundations of LNT, but I can tell you, all of it came out of my story on Hormesis, because my story on Hormesis is saying you got it wrong right from the very beginning. I have an alternative model, and I'll show you my alternative model. So so it becomes a much bigger story when you combine the LNT story with the Hormesis model and with contemporary biology. And the Hormesis model not only is affecting environmental risk assessment for chemicals and carcinogens, but it's really affecting all sorts of how we treat Alzheimer's disease, Parkinson's disease, slow down aging, many different things, and also for commercial products. Hormesis actually affects how drugs can speed up hair growth. It can do all kinds of. It affects everything that a cell can do that relies upon a dose response, which is everything. So I'm sitting on a major driver here within the biomedical community, as well as with regards to the environmental community. And people haven't focused on this, the fundamental nature of how this all fits into an evolutionary perspective either.
And the cell repair stuff, I think, is, on a different side of the cancer story or narrative, is the notion of, the tumors that resolve themselves, right? Or the cells that repair themselves. And so when we have all these early screening techniques that are picking up, we see this with breast cancer, the in situ breast cancer, which a lot of people think may have the ability to resolve itself, right? That very rarely does it metastasize, and people only die from metastasized cancer with the exception of a couple of localized cancers, the brain being the primary one, which is just a matter of space, really. And I think when you think about this stuff in terms of how so much of our, money, energy, research goes to early screening, I think part of the fallacy around that is that there may be a lot of cancers that we're catching early that would self-resolve. And we're like very, I think, naive in considering that approach, maybe in part because of some of this legacy.
Interesting that you mentioned that, because one of there's a lot of studies that support this, but people have tried to take a look at, they call it the multi-stage model of carcinogenesis. And so people have taken a look at, you give highly mutagenic substance to mice, You can look at, liver mutations or any organ mutation, right? And you can see, wow, this mutation occurs and it's a linear fashion, and then you can follow it over time, and then eventually what you have happen, even though you have a linear dose response for mutation, by the time you get to the actual tumor response, you actually have a threshold. So it's exactly that you're getting a lot of repair, and the repair is actually taking place at the low dose zone. And when you have very high doses, you overwhelm the repair capacity. And so this has been shown, repeatedly within very good animal model studies for well-known carcinogens. It's exactly what you're saying, except it's coming in not from a human side, but it's coming from experimental side, well laid out. And that's data that's ignored. I mean, people just they ignore, they don't, what doesn't fit their paradigm.
Well, and it goes back to the human, like the sort of fear-based mentality that I think our culture thrives in, right? Where like you get diagnosed with cancer and you feel you need to respond. And the response of standard of care with chemo and radiation, we actually know causes cancer, right? And damages the cells and probably makes it far harder for them to repair themselves. And so downstream, what is the effect of all of that? And what would have happened if you had, I mean, thyroid cancer is another great example that often self-resolves, they say, right? There's a lot of research coming out about that. And so if we treat thyroid cancer with chemo and radiation, where the cells actually might have had a chance to repair on their own, had it not been caught, unfortunately, right? And then we start doing all these early screenings and we start intervening earlier. I mean, one of the things that I get really hung up on is the survival rates and how we're playing with these numbers, because if we're catching it earlier, right, then your chance of survival is going to be greater. So if I get diagnosed with cancer very, very early, my chance of a 5-year survival is greater even with no treatment, right? Just because if you caught it 5 years later, it's going to probably be more advanced or you won't find it. And so I think there's a lot of this that is, I think, hard for people to understand, because you're really untangling some preconceived notions that we've believed to be certain that are not certain, or where the data wasn't really there, or an assumption was made about some, to your point, like with this, it's an observation that's attributed to mechanism, or we're associating cause with something that's not causal, or that we clearly don't understand the cause of. And we're identifying one variable that's an output, and then all these other systematic things go into play. And it's almost like a self-fulfilling prophecy where like you're going to create the cause that you wanted because of all these sort of fear-based interventions that we're pushing on people. Does that make sense?
Well, it challenges people to go back and be more careful, to be more objective, and not to jump to conclusions too quickly, because jumping to conclusions too quickly can lead to just inappropriate treatment. And I didn't talk about this, but if I looked at my plant situation over the first week, I had a linear decrease in growth, and the second pretty much the same. But by the third week, at low doses, the plants are being stimulated, and it was an overcompensation stimulation. It was exactly what you're saying: is that the plants were damaged. They detected the damage. They attempted to compensate for the damage, and they modestly overcompensated, which is ultimately reflected in this 30 to 60% increase.
I mean, this is also exactly how muscles grow, right? So like the fibers rip, there's a lot of damage, and then it's the repair that builds the muscle.
Yeah.
So in some facets, we are very comfortable with this, and in other facets, we think it's like some other biological adaptation.
Well, it's so you're getting these muscle stem cells that actually migrate into the damaged area, and we've done a paper on that in Hormesis as well, and so forth. Yeah, the body, it's an amazing entity, and it's and I think people like the regulatory agencies, and it's really the public health community, which is a fear-based community. They're not tied into evolutionary biology. They don't understand it, and they decouple from it. And we're here, as I've said before, because we're not sissies. We're actually the result of a long-term, very combative evolutionary process. We're survivors, and to survive You take hits, and then the survivors repair the hits, and they maybe can even use the hit to give themselves an advantage. It's it's a very dynamic evolutionary process, and the whole base of it is that even if you have a damage, um, you can repair that damage. You can repair that damage with an overcompensation that gives you an advantage. Uh, and this is so foreign to groups in EPA in their definition of what is a risk assessment and what it shouldn't include. EPA very explicitly says we will not ever consider the possibility of adaptive responses and repair and so forth. So they're not really interested in enhancing human health. Um, they have a different other agenda, and they have the wrong agenda. The goal is to enhance human health and longevity, the health span, the lifespan, and they want to do something different, and they really need to be redirected, and they got it wrong. Thank goodness. Their hero is really the basic thinking of Muller. Yeah, and I'd admit he's a brilliant man. He had a lot of talents. He's a very hard worker. I have a lot of respect for what he tried to do. I really do. On the other hand, I've identified about 10 big mistakes that the smartest guy in the room made, and the smart people can make mistakes.
Yeah, but the problem isn't the mistake. The problem is recognizing the mistake and learning from it, right? And that seems to be his fatal flaw.
Well, that's true. He was motivated by factors that drove his science rather than him following uh, his science in certain areas. Some ways he could be a very objective man. In other cases, he had the blinders on, and those happened to be when it came to these regulatory issues. I've read a number of books about some have been totally on Muller, some have had certain aspects of Muller, and I find this interesting. People who did deep dives on Muller, very deep dives on Muller, people that I could have learned a lot from reading what they wrote about this guy. Right? I can tell you that none of them who knew about Muller ever found out anything that I found out about Muller. How is this even possible? People who knew Muller for deep dives into Muller's life. I have found out an awful lot about Muller, an awful lot. Published many papers on this guy. None of what I have published on this has ever been reported in these biographies about Muller.
That's interesting.
It's like these writers have had blinders on as well.
Right. Or they're following I know this from working in the media.
Yeah.
When there was a big murder and I would show up and, or there was a woman who won the biggest, this is actually a probably better example because the murder stuff can be really complicated because certain lawyers and you can get in with them or detectives in the same area you've been before or whatever. But there was a woman who won the biggest jackpot ever and she was in Massachusetts. And I had stopped working for 2020 in primetime, but I knew everybody at Good Morning America, and they knew I was in Boston. And she was like half an hour from my house. So they called me, I'm not working there anymore, but they know they can call and that I can handle it. So they called and they said, hey, can you go and just try to find her? And, we had crazy databases where you could find any property anybody owned, any relative, stuff that the average American probably doesn't want to know that the media can get on you in a second if you're at a big outlet. And so I drove out and everybody in the media was hanging out at this gas station, which was where the winning lottery ticket had been sold. And so I went and I kind of said hi to everybody and I met the crew from Good Morning America who was there. And, they hadn't released, or maybe they must have released her name, but they hadn't really said much else about it. Now, this is a little town, right, in Western Massachusetts. I went to Smith, so I'm familiar with the kind of area out there a bit, right? And so everybody's at the gas station, and I was like, I'm just going to go drive around, right? Like she's not going to come back to the gas station. Everybody's got their shot of the gas station, but I want to find her. And so people were going and knocking on doors, right? That was what a lot of the other big network people were doing. So they would go to her sister's house or her kid's house or whatever and sort of beg for an interview and explain how they were the best outlet. I was like, I want to go where no one is. So I went to the diner in town, right? Now, a diner in a small town in Western Massachusetts, they know everybody in town. So I sat and had coffee and talked to everybody in the diner. And sure enough, one of them was like, oh, I'm one of her best friends. I'll introduce you. Right. That only happened because I went the opposite direction.
Right.
I didn't follow the crowd. I went, and where would I go in a small town to hang out and meet people who know this woman? She's probably not friends with the guys that work at the gas station, right? But at the diner, they'll know everybody who knows all the gossip, and they already know where she is and where she's hiding and what she's like. So I think you did a similar thing. It's like the people who are writing the biography, they're just writing based on what everybody else has said before. And not to diminish them, and I haven't read the biographies, but my hunch is with a lot of these things, you think you know the story, and that's the story you want to tell, and you want to tell a better job of that story. You're not looking for a different story. You're not looking for something that unravels the current narrative. And so my hunch is that you came at this because of your, interest in hormesis, and how that completely contradicts the LNT.
Yes.
Right.
Yes.
They're opposite theories. And so you're looking at it from like, how is this guy winning these awards and so celebrated? And we've based all of this information on this, when actually, when you look at the empirical evidence, it doesn't suggest that that's right. So what are we missing in this puzzle? Or what are the building blocks that built this story? And what's the other story? And so I think you've gone in it looking for the other story. And what you found has been, it's profound. I like to joke with people that oftentimes when somebody says, like, how does it feel to be an overnight success or sensation? That 99.9% of the time, the person's like, I've spent my entire life looking at this problem like this, or trying to accomplish this. It's a lot of work. It's not an overnight sensation. But I feel like you're sort of at this moment where you have actually put this all together. And it's really extraordinary, the amount of work you've put into it and the diligence behind, getting the records from the mistress. Like, I love all of that, right? You're trying to really put the pieces together that have been missing. And so I feel like, we should probably wrap up, but I would love to come back to you again and talk to you about hormesis.
I'm going to give you something that I can probably get for you to see. But this is very weird, extremely weird, but it's current. 2 years ago, in July, more or less, I got an email from a woman who is the executive director of a left-wing environmental group that I would normally never interact with, ever. Don't even know who they are. And she writes to me, whom she's never written before. She doesn't know me from Adam, and I don't know her from Eve, really. And she says, I just got this gigantic FOIA that I was from CDC. And I was looking at certain government people, and she said, and your name came up. It was 1,300 pages, and your name came up in about a dozen pages, and you're absolutely trashed in this FOIA, in the email exchanges and whatever they have. I said, so I want to have a telephone conversation with you, because what are you talking about that I'm trashed, right? And so we spoke for about an hour. We probably come up from different ideological perspectives on environment and so forth. Who knows, right? But she sends me all 1,300 pages of her FOIA. And so I look at how I was trashed. I was trashed pretty thoroughly that's where I found out, how I got disinvited and everything else.
Oh, interesting.
That's how I began to find that out, at least in part. But I saw that there were all kinds of plottings and schemings and political machinations and attempts to overtake the Health Physics Society by people who were ideologically supportive of LNT and try to exclude this and whatever. It looked pretty questionable. So what I did was I called up the president of the Health Physics Society, and I said, would you want to see this FOIA? And so ultimately what happened was that I said, well, look at what she got with FOIAs. I then decided the FOIA could be my best weapon. So I then identified about 15 people that I wanted to FOIA, and I became like a FOIA crazy guy. And then I got a lot of stuff on people that were part of this whole cabal, and ultimately all what I got and what the original thing she sent me became a massive article in this, and you won't want to see it. It's on this website called Junk Science, and it makes it very contemporary. All the political machinations that's going on within EPA and within CDC and other people. I come out, of course, being attacked and everything, but it's really interesting to see how you're perceived. And now this article, an article based on that, has just been accepted in the journal called Health Physics, and so it's in press right now, and it's part of the story. It's a very contemporary part of. Has it changed since the Mueller years? No, these people are they are the identical twins of Mueller. They're running the show, and they will do whatever they can do to, you might say, minimize my efforts and prevent any kind of progress that you have, and stop any awards that you might have, any invitations you might have, and 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 I can tell you, as a result of me doing this for you, I mean, say at my age, I don't really care what happens. Actually, I learned that the most recent National Academy of Sciences committee had to take a vote on whether to cite Calabrese or not, and whether to ever use the word Hormesis or not. So they decided not to cite me and not to use the word Hormesis. And I'll have to tell you, in 2001, a National Academy of Sciences committee paid my way to Washington to give them an hour and a half's talk on radiation hormesis, which I did. Now, 25 years later, this National Academy of Sciences committee won't cite any of my papers and won't use the term that the previous committee 25 years ago wanted me to go down and look at. And today we know so much more about it.
Also, I just find it odd that there's like a decree.
Yeah.
Right? That we will never use this word. What, like, that's bizarre.
And at the same time, the scientific community has gone from using it 10 times a year to over 20,000 times.
Yeah, right. So it's not really working out. I mean, the way that they hoped.
This is all part of your story. And it's a complicated story to tell. Because, and it's, that's why I have to say it, when the Health Physics Society came out to interview me, right? They said, "Oh, we'll be there for 2 hours," and they interviewed me for 14 hours, and it led to 14 hours of questions. You can see you and I talking. We could go for 14 hours pretty easily, right?
Yeah.
And but they did it over 2 hours, and then it got condensed into a 10-hour. Health physics documentary, right?
Well, we're going to link to that. I think that's really great for people who want to know more about the story, because we actually didn't really get we probably got about halfway.
Yeah.
And it was superficial compared to the amount of work you've done. So I really do want to.
Yeah. And so we can probably stop now and then go back whenever you figure it out after you listen to this thing and where things are going. And now you have a sense of who I am.
Yes.
And this is my personality. It's not going to change from now until the next one.
Well, I think it's great. We need more personalities like you. I hope that's what this show inspires.
So I'm very comfortable. I'm really appreciative of all the work that you've done and your experience and just the way you do it. So it's actually great. And so I'd look forward to a second go around or whatever it's necessary for you to do what you have to do that makes your work product the way you want it to be.
I appreciate that very much, and all the time you've given me and everything you're doing.